Welcome to our second session of the year of the Signals Research Suite Quarterly Connect Community Series. We released Signals Research Suite last year as a complete unified SaaS software for scientific research, and in the second Signals Research Suite Quarterly Connect, we continue to develop exciting new capabilities for you to use with Signals Research Suite. My name is Chris Stump, and I'm a product marketing manager for Signals Research Suite and Signals Synergy at Revvity Signals, and I will be your moderator for this session. We have a fantastic webinar for you today, but before I introduce today's speakers, I want to share why I think you'll find this webinar useful. So what are you going to learn today? So we assume that you already use Signals Research Suite or Signals Notebook, or you're thinking about doing so in the future. This webinar series is designed to help you unlock more value from Signals. In today's webinar, we introduce you to Signals Synergy, a new SaaS-based solution that streamlines drug discovery CRO collaboration. Signals Synergy adds fit-for-purpose CRO collaboration capabilities to Signals Research Suite and Signals Notebook. We will give you context on why we built Signals Synergy, the challenges it addresses, and give you a glimpse of its key capabilities. We then hope to have some time for Q&A at the end. For this Connect session, we're going to do something a little different. I usually only introduce the speakers, but this time I will co-present with Heather Hayes. I already introduced myself, so I'll introduce Heather now. Heather Hayes is our Signals Synergy product leader. Heather has a PhD in biomedical engineering from Georgia Institute of Technology. She has been leading our efforts in developing Signals Synergy at Revvity Signals, and that's what she's going to talk about today. Before I start us off with this presentation, I just want to give you one housekeeping reminder. This webinar is being recorded, and the recording will be shared with all attendees in the next couple of days. So without further ado, let's jump into the presentation. So today, Heather Hayes and I are excited to introduce Signals Synergy, our new product launching this year. It's designed to boost collaboration between sponsors and contract partners in drug discovery. So before delving into the reasons behind the development of Signals Synergy, I'd like to note the screens Heather will present are from the product's development phase, and therefore the products, the final product you receive may look different. So the motivation behind developing our Signals Synergy platform stems from the evolving landscape of drug discovery. Over the past 15 or 20 years, researchers have been working with a broader spectrum of molecules, ranging from small molecules to nucleic acids, peptides, microbiomes, and stem cells. These diverse drug modalities are not only harder to represent, but also becoming more complex, necessitating greater collaboration across scientific disciplines. The complexity is further amplified due to the intricate nature of the biology involved. So to manage this complexity, we're developing a next generation informatics platform. So Signals Research Suite is an end-to-end solution designed to handle both traditional and new drug modalities. It includes workflows for managing these entities and supports the scientific disciplines involved in the drug development. The suite begins with data capture and collaboration facilitated by Signals Notebook. It then proceeds to data processing, and for example, assay data analysis. And then it concludes with data-driven analytics. And another example would be lead optimization. And so this process can iterate as many times as you need to advance the candidate. So this is a SaaS platform. It's a unified platform and enhances collaboration. And so it increases this crucial. It's basically a platform that we can leverage in order to bring Signals Synergy to allow biopharmaceutical sponsors to incorporate CROs into their ecosystem. And Signals Synergy can make use of this environment in a couple of different ways, and I'm going to illustrate how that works in a couple of slides from now. So another approach to managing the complexity of the drug development from a pharmaceutical and industry perspective is outsourcing. So this strategy is particularly useful when in-house capabilities are lacking or when there's a need to reduce costs and improve operational efficiency. Outsourcing to CROs has been a practice since the 1940s, initially for clinical trials. This expanded in the 90s and includes intermediates and building blocks. And then over the last 15 or 20 years, there's been a kind of a focus or a shift to drug discovery CROs, and looking at this deep expertise for these various drug modalities and access to cutting edge technology, which may not be available in-house. So this allows, an option for organizations to leverage these capabilities. And then when working with CROs, there's an informatics issue or communication collaboration challenge, and often it involves interactions, so the sponsor and the CRO are interacting through spreadsheets, documents, PDFs, and emails with multiple CROs involved. That means that each CRO may have their own kind of style guide for the reports that they provide. And so this complicates the process of converting that information when the sponsor receives it back into a format that's suitable for downstream analytics or even AI or machine learning. And one of our customers on the customer advisory board for Signal Synergy commented to us that they have that they receive slightly different spreadsheets for each one of their CROs, and so they don't really have the staff in order to do the manual curation of the assay results to come back in order to work it up in the Signals environment. And so they were asking us if we could do something about that. Another customer on the customer advisory board was commenting that when they receive these reports back, about 25% of those will have some type of error, whether there's an omission or there's transcription error, and they'll have to send that back to CRO and get that corrected. So we are basically considering these in our development so that we can make this data handling more convenient, because it's not just about convenience, it's actually becoming a business critical aspect of working with CROs. So I'd like to take a deeper dive and just look at the current state of the kind of the communication, the collaboration challenges when working with a CRO. So there's like 4 or 5 roadblocks that we have articulated by talking with customers. So the first challenge is sharing information with the CRO, which requires setting up the proper file permissions and sharing scenarios with your IT organization. And then once this is done, you can start your project and share your spreadsheets and samples and documents. But there's another issue, another roadblock essentially of sharing information, and that is that you can either overshare or undershare. And when I say overshare, you can share, maybe some people in the organization may share proprietary information that you really don't want to as an organization. And then undersharing, because email is being used, sometimes you can email somebody and forget to include them, and actually they're critical to actually initiating the project, because maybe you needed to send them a critical critical attachment and you forgot to include them. So there's that issue. Now, assuming that the CRO gets the information and they can start their work, the sponsor kind of loses track of where the process is. And it would be nice to kind of understand where the process is, the sample or the project is throughout the process of working with the CRO. So once the CRO has actually done the work, then the CRO will copy and paste their information into their style guide, their report, and send it back. It's another potential roadblock in that, there's potential for errors and transcription errors and things like that. And then once the sponsor receives it back, it may not be in a format suitable to do the downstream analytics. So somebody has to write custom code to process it so that the sponsor can figure out if this is actually a candidate that can move forward or not. And so what we're proposing is a streamlined future state utilizing Signal Synergy, where the sponsor and the CRO work within the same SaaS environment. And the sponsor can see all the data while the CRO only sees what is necessary to do the work. So the process begins with the idea, and then it follows through the similar process as before, where the work a CRO is onboarded, start a collaboration at a specific CRO, requesting the work and being able to track that work throughout the process at the CRO. The CRO does the work. They do the make and the test phase, and then they copy and paste the information into a suitable format, but the system does a transformation automatically to make sure it's suitable for downstream analytics. And so then you can return back and understand whether the idea is suitable for moving forward or not. So with Signals Synergy, we have a fit-for-purpose CRO capabilities to Signals Research Suite and Signals Notebook. So Signals Notebook serves as your gateway to the system. And so if you're using Signals Notebook, you can use it with Signals Synergy. And if you need that comprehensive make, test, decide functionality of Signals Research Suite, you can also use it with Signals Synergy with Signals Notebook within the Signals Research Suite. So we aim to address the informatics, the challenges of information management, collaboration, project management, and outsourcing drug discovery research within a seamless ecosystem. This enables drug discovery organizations to leverage external expertise for complex drug modalities, cutting-edge technology. And also, Signals Synergy empowers drug discovery organizations with more insight and less oversight within a single platform. By more insight, we mean that you can focus more on the science, and less oversight refers to the project management capabilities within our Signals Synergy, allowing you to focus less on project management and more on science. So Signals Synergy is designed to address key informatics challenges in drug discovery, and these include prioritizing and communicating work, onboarding and offboarding of CRO resources with a high flux of users, tracking outsourced projects, preserving the sponsor's intellectual property without limiting relevant information, and standardizing and integrating external data for comprehensive downstream analytics. So those are the challenges that we're addressing. And the way we've done that is with these 5 capabilities, these 5 key capabilities. And this is a part that Heather is going to talk about and show you what this looks like in the software itself. So we are providing administration capabilities, so a simplified project initiation, user onboarding and security setup, drug design. So there's an ideation workspace for capturing drug designs and hypotheses, IP protection, so built-in masking of proprietary codes, properties, and material IDs, project management, scientifically minded tools for tracking collaboration progress, and data exchange, so automatic transformation of unstructured CRO reports into structured data ready for analytics or even for AI. So, so these are the main capabilities. And now what I'd like to do is turn this the presentation over to Heather Hayes. Heather is a Signals Synergy product leader, and so she'll show you what this looks like in the software. Heather?
Thank you for setting the stage for Signals Synergy, Chris. Our goal with Synergy is really to address the customer needs that Chris just presented and bring the solution of Synergy directly into the Signals platform. Today, I'll walk you through the Signals Synergy solution that we've developed and how the product can really enhance collaboration and accelerate that collaborative drug discovery process all the way from the idea stage all the way to IND. So I'll be showing you the product kind of screen by screen and walking you through how we view it meeting the needs of the customers. So as Chris mentioned, one of the biggest challenges we see that sponsors face is administering these multiple collaborations with multiple CROs. And that's not just limited to CROs, that can be any kind of contract partner, whether that's a CDMO, a CMO, academic partners, government partners, any type of partner that you're working with that's outside of your organization. Each of these requires security setup and user management. We see also a high turnover of scientists at the CRO, whether you're dynamically picking the assays and services that you need, or just due to the turnover at scientists that we see at these organizations. And that really only further compounds the administrative problem of managing the collaboration and the CRO itself. To reduce this administrative burden, Synergy has a CRO repository for easy CRO onboarding, offboarding, and licensing. CROs can be set up and ready to collaborate with a click of a button and turned off and unlicensed as the business needs change. Each CRO has a CRO administrator who can manage their own users kind of in a self-service way and organize them into departments or groups that might represent the services you're requesting for performing specific kinds of work or assays or synthesis work, while the sponsor retains at all times oversight of licensing and what exactly the CRO can see. So we want to take you from saying, with the personnel turnover at CROs and the many CROs we work with, administering CRO projects is a huge burden, to Signal Synergy makes it easy to initiate new collaborations with our favorite CROs or whatever our needs are at the time. And the CRO admin helps me manage their users so I can focus on the project and the science itself. So after setting up a CRO in Synergy, the sponsor can easily initiate a collaboration with the CRO. I think of a collaboration as representing a dedicated secured slice of your tenant for that CRO to work in. You may have multiple slices for different projects or collaborations that you're undergoing, but these slices represent, or these collaborations represent, places where you can exchange work, record experimental records, and capture and gather and transform data for a specific drug discovery project. When you start with a new collaboration, you can first assign it to a group of internal users who are going to manage that. It can be multiple different managers from your internal side. And then you also assign it to a CRO. And you can pick and choose which of the groups within a CRO need to work on this particular collaboration. And that can be dynamic over the course of the collaboration. Importantly, our customers rely on attributes or fields to help organize, secure, and search for their work. However, we found that some of these attributes may be proprietary. Maybe there's a hot new Alzheimer's disease protein that you've discovered that's a target for this particular project, or maybe you have top secret project codes, or maybe there's material IDs or other information related to this collaboration that you're not working on with this particular CRO. And you may want to hide those fields from the CRO while retaining that ability to organize and search for all of your work, whether it's internal or external, by the same attributes. So we've added the concept of internal fields, which are hidden from the CRO. And sponsors will be able to add these internal fields to their collaborations and other Synergy entities like their experiments that are hidden from the CRO. So on the sponsor side, these fields are visible and searchable by the sponsor, but on the CRO side, they only see exactly what they need to see, whatever it is that you want to share, while protecting any kind of information that you don't want to share. We really want our administrators to feel that they can organize and search for their internal and external work with the same attributes without revealing proprietary information. And they can share as much or as little as they need to successfully execute the project. And this can be dynamic, project to project, and over the course of a collaboration. So where does a collaboration typically begin? It often begins with an idea. Ideas in Synergy are a new entity for capturing and tracking sponsored drug ideas. Each idea represents a hypothesis about how to make or improve a drug. And inside an idea, sponsors can propose drug designs or new design sets for their small molecule compounds or their biologics. These designs are like sketches on a back of a napkin. They're proposed structures or sequences that you think might work and might bind well, might be soluble, might deliver the active drug in the right concentration. And some may be successfully synthesized and progress to 2 leads, but many others may fail to even be synthesized or may fail quickly in a primary assay. So there's no need to clutter up the material registry with them. So in that sense, designs are like materials, but they are quite different. Designs may not have been made yet, and so they may not have an amount or a batch or an appearance. And so designs offer you this chance to capture this idea and then track it as it progresses. You see that we can pull in different properties that are predicted off of this sketch. In addition, we can track the various work orders that have been sent out in relationship to this design. And eventually we might even track the registered material that came from this design. Designs have statuses, so when the lead is progressing, you can keep it in the active state. When a lead has failed, or maybe it's failed at synthesis, you can close this design and note why, so that later down the road, if you propose a similar design, you can go back and look. Importantly, this table is prioritizable. So when you learn more information, whether that's from predictions or in silico modeling, or maybe from the work that's been sent out, you can prioritize the designs that are becoming your top leads. And so as you gather this data, you can really identify the ones that should go forward first and are most likely to succeed, while removing the ones that are likely to fail. So the purpose of ideas is to really provide you that place to capture your ideas and track the progress of the leads down the scientific pipeline, and take us from just a make, test, decide to really a design, make, test, decide. Having that starting point, all those ideas, all those designs that are coming into Signals, one place of truth to record that and take you then from the design, make, test, and decide as you track those leads through the pipeline. So now that the sponsor is ready to share the designs with the CRO, maybe for synthesis, where do they begin? They begin with a work order. So now, importantly, as I show you the work orders, they don't have to start at an idea. They may start much further down the pipeline. They may start with a registered material, maybe a sample sitting on the shelf in a vial with a barcode that you're ready to ship out for some high throughput screening, or maybe ship out for formulation to go into preclinical animal studies. So work orders don't have to start from here, but when they do start from here, I'll show you what that looks like. So once you've gathered your designs and prioritized them, you can then set up a group of top priority designs and send them out to a work order for a CRO to execute. You'll notice again that all work orders that are sent out are tracked here right in at the design by design level, right in the design table, so that you can know, has this been synthesized before, what assay has been run. So what are work orders? This is here showing you what a work order would look like. It's the way to request work from a CRO, and it also has some key features to allow for secure passage of only the necessary information to the CRO. So first of all, work orders are prioritizable. They can also be group assigned to a CRO department. We talked about those departments earlier, so you may not know exactly who's going to execute the work, but you know what group you're sending it over to, or what the type of work that you're requesting is. They also have an expanded set of statuses to keep both the sponsor and CRO up to date on the progress of the work. So removing the need for emails or other tools, you can comment right within the work order. You can place it on hold and wait for important information. You can indicate that it's waiting on important receipt of materials. Work orders can also contain all the documents, chemical drawings, tables, anything that you would typically contain in a Signals entity, like an experiment. You can contain here in a work order, so you can send over an SOP, or you might be sending over some tables or some proposed drawings. Importantly, each work order is fulfilled by a sponsor-controlled and templated synergy experiment to ensure that the details of the experiment are properly captured and also properly linked to the work order, so that you can have full traceability on the data that was performed. And it really gives the sponsor control for what they're expecting back. One of the most important features of a work order is the ability to control what is passed at a very incremental and granular level. So when you need to share a material or a batch or a design, so anything that's material related, the sponsor can select which properties of that material related entity are shared with the CRO and which properties remain only sponsor facing. They don't have to share the entire entity. We find this very important because our customers use work orders for many different things. For example, when asking a CRO to synthesize a design, they may need to share all of the information. They need to share the arrangement of atoms, the chemical structure, the monomer sequence. They need everything in order to be able to successfully synthesize it. They still might want to predict some predictions about binding to a particular target, but in general, they want to share a lot of information about that design. But if they're sending over some samples for screening, they may want the screener to be blinded with only an ID and basic hazard information possibly. And so reference tables really allow you on a dynamic basis, work order by work order, to decide what you need to share with the CRO and what you don't need to share. And reference tables really only take a copy of that selected properties, and so nothing in addition, nothing beyond what you've selected to share is shared with the CRO. It also puts it all right here within the work order, so it's very easy for a CRO to come in and see exactly what they need to execute on that work. So what does it look like to create a reference table? So first, the sponsor user who is setting up the work order would select a particular material library, and then they might check whether it's a design, an asset, or a batch. And then they're presented with a set of available properties that are shareable. And that's set up by the admin, and they can select which of these to share. As additional layer security, the admin can mark some properties as sensitive. So for example, you might mark the chemical structure or sequence as sensitive. And sensitive properties simply mean that the sponsor user who's creating the work order must be privileged in order to share those into a reference table for the CRO. So again, it's a highly secured level of passage, and the admin has control of how secure they want it to be. Once all of that is set up inside the work order, you'll notice that we have another layer of offering for this externalization of these materials. One of the most sensitive or proprietary pieces of information we find is the material ID itself. Material IDs are often used in patents or publications or submissions to the FDA. They also provide a way to link all the different batches. And so when you run different assays on different batches, it provides a way to link all of that data together. And so in Synergy, we automatically provide the sponsor with an external ID that can be used with external collaborators. In work orders, you'll see from the sponsor perspective, they see both side by side. So in this case, they're seeing a compound batch ID right next to that aliased EX ID, what we call an external ID. But on the CRO side, they only see the EX ID. They can use this to communicate back with the sponsor. It's a deterministic EX ID that points to one and only one batch or asset. And so they can also use this to communicate back the data. So overall, really work orders are intended to make it easier for the sponsor and the CRO. As a sponsor project manager, Signal Synergy allows me to set up to protect my material IDs and choose when to share the compound structure or protein sequence. And a sponsor project manager can share as much or as little as they need to share based on the individual work. And as a CRO scientist, we want them to experience that they have everything they need right within the work order to complete their work. There's no need to go searching through an email to find an Excel list with the list of compounds that needed. There's no need to look at maybe some other project management tool to see which tasks are coming, whether everything is right inside the work order. Along those same lines, we've also empowered Signal Synergy with some tools to simplify project management and to perform project management right within the Signals platform. In this case, we've included Kanban style work queues. So work orders come with this powerful and user-friendly project management tool, these Kanban style work queues that are both accessible to the sponsor and to the CRO. I'll point out a few things about them. They're, of course, organizable by different things. Here you see it organized by status. You can also organize it by priority, filter by the assignee or the issuer, filter by which CRO or collaboration you're working with. In addition, each work order has a color coding, and that's by the work type. There's an ability to set the work type, which identifies the assay or service or type of work that's being done. Maybe it's synthesis, or maybe it's in vitro ADME, or maybe it's all the way into the PK arena. And this allows the sponsor and the CRO to easily track the progress of their leads or designs. You'll see that if there's a chemical structure, that can be centered and previewed right on top of the card, so it's easy to find, oh yeah, where's that assay that I requested on this particular compound, or how's the synthesis going on this particular library. So it's easy to see where your leads are in the pipeline. Are we at the synthesis stage? Are we further along? Track it at the collaboration level, at the CRO level, at the assignee level. And they can be filtered and rearranged as you need to best suit the persona who's looking at them, or best suit the current collaboration landscape. And the CRO also has access to the Kanban board, so they can easily prioritize and plan their work. I think this is very important from the CRO side. Often, because they work with so many different sponsors, they're often logging into many different platforms. And we want Signal Synergy to be the easiest, most accessible, and most powerful tool for them as well. And so when they log in, they can go straight into their work queue. They can easily navigate to the last experiment and work order that they were working on. They can see what's coming down the pipeline over the next week or month, so that they can reserve maybe machine time, or know what reagents to order. It makes it very easy for them to know what work is coming, what experimental details are requested, and to keep track of all their work. And also to keep the client automatically updated. They aren't having to email them and let them know how things are going. As they add the data, the Kanban board is updated. So it makes it very easy for them to communicate within the system without having to go to multiple third-party tools, or worse, using email and spreadsheets. So what does it look like now that we've established a set of work orders? We know that these are fulfilled by Synergy experiments. What does it actually look and feel like to operate as a CRO user here inside of Signal Synergy? And how do we go? I'm going to take you all the way through from logging in and going through a work order all the way to the sponsor registering a sample that has been successfully synthesized for them. And again, this is just one example. Synthesis is just one way this platform can be used. Okay, so the CRO user is here at their Kanban board. They're going to go into a work order that they have in progress. Here inside this work order, they can see the priority. They can see, for example, that it's assigned to them, or maybe they're a manager of this department. And here the sponsor has shared with them 2 different types of reference tables. They've asked them to use these materials. Maybe these are intermediates that have been shipped to them. And in this case, they've shared just the ID so they can communicate about it, as well as just a few properties about it. Whereas the designs that they're asking them to synthesize, you can see that they've shared everything almost. They've shared the chemical structure, they've notated here the amount they want. They could also request a certain purity or delivery method, or maybe tests that they wanted to be run. And they've shared various other properties about these designs. So you can really control what you share, and you can share different aspects of different things, whether that be a reagent or a design or a plasmid, whatever it might be. Okay, and so now they're going to go up to the experiment that has been set up for them and that they've already started work on. And here, just like regular experiments, they can do all the things they normally do. But importantly, Synergy experiments have some fit for purpose special handling. So here you'll see maybe they've added some background documentation. They've also mapped out the reaction that they've performed, created some products, and then created some samples here. So the first fit for purpose thing is that inside the samples table, so the CRO has synthesized these samples, they can use that work order, that related work order, to relate and tell the sponsor, yes, what is sample 13, 14, and 17? How do they relate to what you've requested? So here they're going to use this reference column to look up the various items from their work order that were requested. In this case, it's done some chemically smart matching, and they can show that sample 13 is design 18 that was requested. They can then see that the amount is very close to what was requested. And again, they're just going to relate each of these samples to some of the designs that were requested by the sponsor. After they're done, they can submit that experiment back to the sponsor. And now we're looking at the sponsor side. They can go back and review the work. They can see everything about that design that they related it to. And now they're going to register that sample that's been queued up for them by the CRO from right within the CRO's experimental record. So they're going to grab that information either from the table and begin that registration process. Here on the sponsor side is where the duplicate checking would occur. They might recognize that this is another batch of something that's been previously synthesized. And then that material ID is written right back. They can link out, find everything they know about the material, including batches that were made by other CROs or any other predictions or other knowledge that they have, properties about that material. Whereas on the CRO side, since this is right inside their experiment, what they're going to see is actually just the EXID. And so now they have an EXID to relate this sample that's sitting on their shelf, and they can progress that through other assays as needed. And so even though you're registering from right within the CRO's experiment, the sponsor can retain additional information that is not necessarily available to the CRO, whereas the CRO can record everything they need about the experimental record that they need to communicate to the sponsor. It's all about making it easy to work, but also protecting that IP. Okay, so once the work is completed, the data is a major challenge. How data is shared is often these spreadsheets, these multi-tabular spreadsheets that are communicated via email or possibly file sharing platforms. They are often very human readable and contain lots of rich information about how the assay was performed, who performed it, usually a cover sheet about who performed it, the date that it was performed, which site it was performed at. Maybe details about the materials or assay conditions, various reagents that were used. Further into the report, if in this case it's a plate-based assay, you might see a very human readable plate map where you know your controls are here on the edges, you know that you have a threefold dilution of various compounds. You might have the identifiers for these compounds in a separate table. And then even the raw data, you'll see that it's laid out as a plate We also see some information. For example, you might have asterisks indicating outliers, and you might have indicators for wells that were below the level of quantification. And so while this is beautifully human readable, computer consuming this data that's multi-tabular, where the metadata and the raw data and the organizing information is spread across multiple sheets. Also, clever human tactics or patternings for these type of reports that work great for human readability are a challenge for computer consumption. Things like merged cells or textual embedded headers that show how things are distributed on a plate. It makes it very difficult to extract this data and to bring this data together with the other data you have about your leads and make the most comprehensive decisioning possible. Oftentimes this involves bringing in resources, sometimes even that manual copy and paste that introduces error, or having some dedicated resources that are constantly keeping up customized scripts to extract this data automatically. And so it really is a big challenge, a common entry point for error, and really slows down the process from receiving the data to taking that next iterative action of choosing a different lead or prioritizing different leads or just requesting the next assay. It's a major loss of time and resources. And so what we want to do is solve that within Synergy. So what you really want is to be able to take this report and automatically transform it into structured columnar data tables that are consumable, that you can use alongside your other assay data and relate that data. You want to extract the raw data on a well by well level, so you have that source data, or maybe it's an animal by animal data. And then you also want the measurements, the analytics. So if you're reporting an IC50, you want to be able to capture that analytical endpoint in relation to, for example, the compound batch ID. So what you want to be able to do is take that report and automatically extract the valuable information so that you can move quickly through that cycle and get from that idea all the way to the correct lead very quickly. So what we've done is we've included a feature called the Synergy Data Exchange. It's part of the Synergy project that does this for you. The Synergy Data Exchange is built on top of a robust library of CRO and assay-specific report formats. So importantly, these are the reports that we typically see back from the CRO. These aren't reports that we've specified, but rather every time a CRO sends back, a plasma binding assay or a PK study, they tend to come back in these same forms that have slight human variations that the tool handles, such as the table's been moved, or there's maybe a new comment line inserted or some coloration. But they tend to be fairly stable formats. And so we have a robust library for covering these reports. Each report is stored with a set of parsing instructions that can be sent to the engine and produce out and extract out the combination of the metadata, the raw data, and then analyze. It's part of the Synergy project that does this for you. The Synergy Data Exchange is built on top of a robust library of CRO and assay-specific report formats. So importantly, these are the reports that we typically see back from the CRO. These aren't reports that we've specified, but rather every time a CRO sends back, a plasma binding assay or a PK study, they tend to come back in these same forms that have slight human variations that the tool handles, such as the table's been moved, or there's maybe a new comment line inserted or some coloration. But they tend to be fairly stable formats. And so we have a robust library for covering these reports. Each report is stored with a set of parsing instructions that can be sent to the engine and produce out and extract out the combination of the metadata, the raw data, and then analyze endpoints in an automatic fashion. So to show you a little bit of what this looks like, this is the data exchange element sitting right here inside of a Synergy experiment, just like the experiment we were just looking at. Show you it from the CRO's perspective. They can log in and they can see the type of report that they've agreed on with the sponsor. This is the report they always send back, in this case for cell viability. They can look at a template that they could download to fill out, but most likely their internal systems are already set up to populate these reports. They can see the type of tables that are expected, make sure they've gathered the endpoints that they need, see how the raw data is laid out, possibly how the plate map is expected or the compound table. And when they're ready, they can just simply upload that multi-tabular Excel-based report that's typically emailed. They upload it. And once they upload it straight into the data exchange, you'll see that there's automatic processing. So that's sent out to the engine and they'll receive back a validation status. So if it's valid, they know that the report has all the information that's been requested and expected. Otherwise, they'll receive back a failed with location-based error. So, oh, it's missing this table, or the data type in that particular cell is incorrect. So they receive that status back right away so that they can correct those errors before the sponsor ever picks up that data. And then on the sponsor side, you'll see that they have additional information besides just being able to view and download the report. They also have access to these transformed tables. So here you see, for example, the well level details with various metadata indicating the source file, as well as the analytical endpoints such as the IC50 and hill slope percent inhibition that they can extract from that report. And they also have access to the original report stored right there, all within the Synergy experiment, not spread across multiple spreadsheets, file sharing, or email, but rather the data ready to go for downstream analytics, whether that be machine learning or AI, bringing it into Inventa for SAR-like analysis. These tables are ready to go, downloadable into your data lake, or if you're using the research suite, directly consumable into Data Factory for downstream analytics in Inventa. So the idea is that all within the Signals platform, all within the research suite, you can take that data automatically and make those analytics fast so that you can progress the right leads forward and do so efficiently in a friendly, collaborative platform that's easy to use for both the sponsor and the CRO, avoiding any kind of error introduction, copying and pasting, or the heavy lift of maintaining a set of parsers for these reports on your own. So Signal Synergy is, again, really designed to take you from idea to INDA, no matter the modality, whether that the work is done in-house, externally. It should be a seamless process. We want to provide easier administration of CROs and collaborations. We want to provide a design and ideation space, especially as we see more and more outsourcing. The sponsor really becomes the source for that idea and the need to track those ideas through, whereas a lot of the work is outsourced. We also want to protect IP. We know that that is an extremely valuable piece, and we want it to be easy to work together without constant redacting, it just to be a seamless process where the IP is protected, but everything that's needed by the CRO is available to them. We want to simplify project management. We want to take it out of email, out of other tools, out of spreadsheets, and right inside the Signals platform. And finally, we want to rationalize that very complex data exchange step so that the data, which is really the gold that comes out of here, is readily accessible, readily usable for efficient and comprehensive decision making. So overall, bring you more insight and less oversight with Signal Synergy. It's coming out just within a month's time, so we're very excited to deliver that to you within a few months' time. And thank you for your time and attention, and we look forward to discussing Signal Synergy with you further.
Thank you, Heather. So that was a really nice overview of Signal Synergy, so appreciate that. And I also want to apologize to the audience. We lost power on one of where we were hosting the webinar from just for a few seconds there. It wasn't Heather's fault, so I just wanted to point that out. Okay, so let's get into questions here. For Q&A, so we have, let's see here, let's just start out with this question. So Heather, is Signal Synergy a cloud-based SaaS solution?
Yes, absolutely. Signal Synergy is built right on top of our existing Signals platform. It is a cloud SaaS solution. So you will receive updates for Synergy on a monthly basis, just like you do for our notebook and Signals Research Suite.
Perfect. So another question is, someone asked, because this is Signals Research Suite Connect, they said, they already have Signals Research Suite. Can they add Signal Synergy to their existing tenant?
Yes, that is a very easy process to turn on Signal Synergy for your existing tenant and add licenses for your external partners, your CRO users.
And what about, so they didn't ask this, but what about if somebody's listening from the audience that they have just Signals Notebook, they don't have Signals Research Suite, what's their option there?
Yes, you can add Signal Synergy to a notebook only tenant. You can add it to a tenant that is Signals Research Suite or a mix of either of those.
All right, perfect. So another question was asking, I think when you were on the ideas section, they ask, is there a text field so you can capture why an idea was not pursued? For example, the reagent was expensive, or perhaps why it failed, a synthetic challenge, etc.
Yes. So built into the designs out of the box is a status capability. So designs can be left open and they're a progressing lead, or they can be closed. And as you close that design, there's an option to provide a status reason. So if you go back and propose a similar design later, you can go back and look at that previous design and know why, for example, you closed out that design. So you might there note right within the status reason that's already provided. Like you said, a synthesis challenge, maybe it wasn't soluble enough, or like you said, the reagent might be too expensive. So status and status reason are built in to each design at a design level. And then of course, through the ideation sign and close process, you can also provide a reason why the idea succeeded or failed, or why it was transitioned to a new idea.
Okay, perfect. So we got another question that just came in. So it's kind of related. It was asked by a different person, but it's kind of related to the question about being a cloud-based solution. So where is the data physically stored? Will it be on the sponsor server or on Revity server?
Yeah, so that depends on whether you're a standard customer or an E3 or private cloud customer. But for our standards, the data will be stored on Revity servers, just like for Signals Notebook or Signals Research Suite. The database is completely shared between Synergy Notebook Research Suite. It's all stored together on the back end. Synergy, the difference there is the additional features and the additional user interfaces and workflows, but the database is shared.
Okay, thank you. So another question is about kind of the distinction between the licenses between the sponsor and the CRO. So does my CRO have to own a license of Signals Synergy in order to use it?
So they do not need their own instance of Signals Synergy or the Signals platform, but you as a sponsor need to offer them what we call a Synergy CRO seat in order for them to be able to log into Synergy and work with the features there for Synergy. It's a unique seat license. We keep CRO users distinct so that we can provide the automated and robust security that we discussed. But they do need a license, but they do not need their own instance or tenant.
Okay, that's perfect. Okay. So another question is, if the sponsor already has Signals Research Suite or Signals Notebook, do they have to log into a separate instance of Signals Synergy?
No, it's like I said, it's all on the same platform. And so it's a very seamless movement between the different applications. So many of the workflows connect directly out of Notebook. For example, if you're in search, you can easily pull back your experiments and your Synergy experiments together as one using the same attributes. If you have an idea with your designs, you can look at those in the Notebook or the Synergy interface. If you want to move into Synergy to work with your Kanban boards and track the progress of your collaborations and work, that's easily done through the app switcher. But you're staying all within the same platform. You're just moving from screen to screen or interface to interface. It's one platform.
Okay, yeah, that makes sense. Okay, so I think this is the final question. So this is towards the end of your presentation. Can you explain how Signal Synergy Data Exchange library works? Specifically, is there like an initial library of parser templates?
Yeah, so we are excited to launch with quite a robust library of CRO and assay specific reports and transformers is what we call them. So provided out of the box is a Revity supplied library that covers a large variety of commonly requested assays and assay formats that you can use right at the purchase of Synergy. We have a long-term roadmap approach to the Synergy Data Exchange. So initially, we'll be working with that out-of-the-box library that we are providing for you. You can do some of your own configuration and customization. And with our early adopters, we are happy to cover additional reports in the library so that those are ready to go for you. We will handle the engineering work on that side. And then we'll move into more of like a services type model for requesting additional report coverage. And then very longer on the roadmap, we plan to offer a user interface for customizing and designing your own reports.
Okay, that sounds very powerful. So it looks like that's the end of the questions, Heather. So I want to thank you for providing this excellent introduction to Signal Synergy.
I do want to make one clarification.
Oh, yeah, sure.
So you'll notice there was a slight delay between our initial recording and this presentation, and I did want to clarify the timeline there at the end. I mentioned coming soon. It is literally imminent. So Signal Synergy will be released on 6/15, so June 15th, with the 24/5 release of Notebook and Research Suite. So I just want to clarify that timing. We did the recording a little bit earlier, so I wanted to clarify that.
Yeah, that was a good point. I had forgot to raise it. I think the 15th is actually this Saturday, right?
That's correct. That's when Synergy will be available for quoting and request and going live on your tenant. Okay,
perfect. All right. Well, thank you, Heather. So that was the end of all the questions. So, Karen, would you advance? Yeah. So I just want to thank everybody in the audience again for joining for our second Signals Research Suite Connect community series. So as we close the webinar today, I'd like to share just a few announcements with you. So first, mark your calendars for the third Signals Research Suite Connect. That will be on Wednesday, the 11th of September. And if you liked what you heard here, I mean, this was a kind of an update on a new product release. But in general, if you're interested in learning more about our products and kind of how they work, we have a full range of educational courses from beginner to advanced that covers our Signals family, covers ChemDraw, covers Spotfire, and you can contact Stacy Fellows at the email shown on the screen. And then finally, I ask if you have any suggestions for future topics, or if you have an idea for a presentation or maybe a data set or something like that that you think would be interesting to showcase within Signals Research Suite, contact me via the email that's shown on the screen there, and we'll look about incorporating that into a future Connect. And I just want to thank Heather again for providing this insightful introduction into Signal Synergy. We're all very excited about Signal Synergy. We think that it is very timely with all the sponsor and CRO kind of challenges with data exchange and collaboration. So we're very happy to offer that to the industry. So we also I noted this in the comments that we recorded this session, and we will be sending that out in the next couple of days. So watch your inboxes for it. And this concludes our session for today, and I hope you have a nice day.